by the Department of Biotechnology, Physics, Electronics, History, and Chemistry in association with IQSC. Today, I'm here to introduce the today's resource person, Dr. Ravikiran Yeddi. I would like to introduce this person to all the audience who are present online here. His educational career. He did his PhD from Wayne State University, USA in 2009, MS from Wayne State University, USA in 2003, MSc from Andhra University, Visakhapatnam, India in 2000, BSc from Andhra University itself, India in the year 1998. Teaching and research experience of this great person. He has been involved in various research roles since 2000, including research intern, research technician, research assistant, assistant professor, research associate, scientific advisor, senior scientist, founder, principal scientist, visiting professor, and more. Awards and honors are very common for this person. From 2019, guest of honor at the felicitation ceremony of delivering the public lecture and sponsored by the Department of Biotechnology and organized by Grand City University, Bangalore. In the year 2018, guest of honor at the felicitation ceremony for this person in highly accomplished alumni of Government College organized by the Department of Chemistry, Government College, Rajahmundry, AP, India. He also received in 2017 the honor at the National Workshop on Biomass and Bioenergy as a resource person at Rajahmundry, AP, India. In 2017, this person received an honorary award and for presenting on the research activities and the drug design methodology to the PG students at Geetham University, Visakhapatnam, Andhra Pradesh, India. Fellowships and scholarships were so common to this person, and he received 12 fellowships during his career. Collaborations of Dr. Eddy he had collaborated with the 12 institutions for research and development. Monetary experiences. He has monitored over 500 students in the span of just 17 years. And invited talks. He has received more than 25 invited talks since 2008. Oral presentation. He has more than 50 plus oral presentations since 1998. Publications. They are in N number of, and we consider the international publications for research. And he has authorized 26 in different reputed journals. Patents. He holds 5 patents. And it says Dr. Eddy has a strong academic and research background with numerous achievements and contribution to his field. Now I request Sir Ravikiran, sir, I hand over the program to you, and we are eagerly waiting for your presentation here.
Thank you. Thanks for the introduction. Can you guys hear me?
Yes, sir.
Trying to share my slides. Just a second. Can everyone see my slides?
Yes, sir.
Yes, we proceed.
Okay. Good evening, all. I am Dr. Ravikiran Yegudi, I'm the founder and the principal scientist and the instructor in the Center for Advanced Biological Sciences and Entrepreneurship. We abbreviated it as TCAMC. So TCAMC is a startup company that I started in India after I came back to India. So thanks for the elaborate introduction. So as you already know, that I have done my master's and PhD from the United States, and I worked as a U.S. federal government in Washington, D.C. And I have a startup company in Canada, in Toronto. So my startup company in Toronto works on Alzheimer's diseases. So we are trying to identify new drugs for the Alzheimer's. So the TCAMC lab in India, we initially started in Rajamahendravaram, and then we slowly moved on to Visakhapatnam. So in Visakhapatnam, we are actually located within the campus of Andhra University. So my lab is located, we are incubating in the Andhra University Zoology Department. So that's where we got the lab space for our lab. And I'm very happy. I have to thank my senior, Dr. Shambhav,
So we have some queries in the chat box.
for giving me this opportunity. Can you hear me? Hello?
Sir,
Can you hear me?
there are some queries in the chat box that your voice is not clear. So please adjust your mic once, please.
Well, yeah, let me check. Hold on just a second. Can you guys hear me now?
Yes, sir.
Okay.
Thank
All right, so we started the TCAMC lab in the Department of Zoology recently. And initially, this was started back in 2019 in Rajamahendravaram. And we started the branch in Visakhapatnam in 2020. And now we're celebrating the second anniversary in the campus, Andhra University campus. As I was saying, I'm happy and I thank my senior, Dr. Shambhav, for giving me this opportunity. Thank you, sir. So I'm actually glad that the topic for this webinar series is on the research methodology, because when I came back to India, the one thing that really bothered me was the way how research is done in India. Yes, you're doing an experiment. Obviously, when you do an experiment, you get a result. It could be any experiment. But the problem is, how do you analyze the result? How do you analyze the data that you get from the experiment? That's very important. So let's say, if you're talking about a person, say, oh yeah, that guy is tall. Okay, that's fine. If that guy is tall, how tall is that guy? Is he taller than somebody else? Or is he taller than a tree? Or is he taller than a building? Right? So if you don't tell the people, then, it doesn't make sense. Because in science, we believe strictly in the evidence. It's evidence-based knowledge. So we compare things and we understand. And as you know, we know the details of a lot of things at the atomic level. We are doing molecular biology, in which we do different types of molecules and the mechanisms and the cellular biochemical processes. And not only that, we actually go down to the atomic level. So we know exactly the length of the chemical bond, a covalent bond between two carbon atoms. Or we know the bond angle in the water molecule, right? So we are actually able to look at things at the atomic level, which means, it's a great thing. And, you have to do things systematically, because science, you cannot just do the experiment and say, I got the result. What does that result mean is very important. So that's why we have to use proper controls. So today I decided to talk about how to use different controls when you do experiments in biology or life sciences, so to speak. Okay. So there are different types of controls that we can use in the experiments. So depending on what kind of experiment you're doing, if you belong to physics department, your controls are different from, if you're from a biology department. And, if you're a mathematician, your controls will be different from, somebody who is doing political sciences. So there are different types of controls, but broadly speaking, you can divide them into two controls, two types. One is the positive control, and the other one is the negative control. So the positive control is something that would, it is also an experiment, but you already know the result, okay? So the control experiments are something that you already know the result, but we do them anyways because, for example, in the case of positive control, we compare the actual experimental result with the positive control experiment. So you can measure the quantity. Okay, so let's go back to the example that I gave you. Let's say you're talking about a person. You say he's tall. So when you say he's tall, is he taller than someone, or is he taller than a building, or is he taller than a tree? So these are the things that you have to, use. So that is a positive control. So you have to make sure that you have a positive control every time you do experiments, whether it is molecular biology or microbiology or, computational chemistry or anything. When you're doing science, controls are important. Anyways, and the second type of control is the negative control. The negative control is something that tells you whether your experiment is working or not. So that tells you the quality. Okay, so did it work properly, or is it dirty, or something like that. So there are two types of analysis that we do. When you do an experiment, when you get those results, you look at those results, you perform two types of analysis. One is quantitative, and the other one is qualitative. Qualitative means, did you get the result that you were expecting? As in, did the experiment work, or did the experiment fail? So those things are the qualitative analysis. Quantitative analysis is the quantity, as in, if your experiment worked, how big is the result? Or, how tall is that human being that we are discussing about? So it's quantitative, you're measuring the quantity. And if somebody shows you data without controls, please do not trust the data, because without the controls, it's hard to estimate. Just keep in mind the example that I gave you in the beginning. When you're saying, somebody's tall, and if you don't give the comparison, if you don't give the positive control, then people don't know how tall that person is. So that's why controls are very important, and you should always provide your experimental data along with the controls. Sometimes it's the best experiment always has both positive and negative controls. Sometimes, we may not be able to do both positive and negative because of various reasons. For example, in labs, sometimes when you don't have enough funding, then you may not have enough money to buy these chemicals and reagents. What you can do is you can do just a positive control or just a negative control. But if the experiment that you're doing is something that is novel in the sense you're the first one to do this experiment, then you must have both positive and negative control. So, I mean, without controls, you cannot look at the data. You should have at least one control, either positive control or negative control. The best data is always the data that is presented along with both positive and negative control. So that's why you have to be careful. Okay, so now I am going to pick a few examples of these controls. I think most of the life sciences students, you might already know what this agarose gel electrophoresis means. So this agarose gel electrophoresis, this is a technique in which we use a gel. So I'm showing you the gel here. You can see this rectangular piece that is shown in this picture. That is the agarose gel. So it's called agarose gel because it is made of a carbohydrate called agarose. So within this agarose gel, we have these small wells into which we add different DNA samples. So now, as you can see, there's a DNA ladder in the middle. So this is a DNA ladder where this is a sample that has multiple bands. So let's call each of these horizontal lines as bands, okay? So in this particular well here, we have loaded something called DNA ladder that has different bands. And the importance of this DNA ladder is that we know the exact molecular weight of each of these DNA bands in the DNA ladder. So we can use this to compare our samples. So on the left side, we have a few samples from a ligation reaction. So that is one type of experiment where we monitor of DNA. And on the right side, we have a restriction digestion reaction where we take DNA and we cut it into pieces using different enzymes. For example, here, we are using a control here that is HIND3. So HIND3 is the name of the enzyme that we used to cut this particular sample here. Okay, so this is our positive control. And so this is a positive control here. And so the DNA ladder that is loaded in the middle will provide these different bands of each molecular weight so that you can actually compare your bands. So let's say all these samples on the right side, those are your DNA samples. And let's say if I want to know the molecular weight of this band, particular band here, then all I have to do is compare which band in the DNA ladder is it coinciding with. And based on this molecular weight, I can actually estimate the molecular weight of this one. So that's the purpose of the DNA ladder. So before going further, let me just briefly tell you the differences between ligation reaction and restriction digestion. So in a ligation reaction, what we do is we take pieces of DNA and then we add an enzyme called DNA ligase. So when you incubate this reaction at 37 degrees or 15 degrees, depending on the type of DNA ligase enzyme that you're using, all the pieces of DNA will be joined together into a single piece of DNA. So as you can see here, you can see only a single band. Here, a single band, and this is a single band, right? So looking at this, we can definitely, confirm that the experiment worked, right? And on this side, you can see the restriction digestion, where we took a DNA and we cut it into pieces using different enzymes. And so the final product that we're expecting here is that single piece of DNA, which should look something like this on the left side, becomes like this with multiple bands because it has been cut with the enzymes, right? But now let's say I have 1, 2, 3, 4, 5, 6 samples here. And let's say I forgot which sample was cut with the enzyme called HIN3. Okay, so in order for me to do the analysis, what I did was in this particular well here, I purposefully loaded HIN3 digestion, or the DNA that was digested with HIN3. So I know for sure that this well has the HIN3 digestion. So if you look at the banding patterns, you can see that there's a thick band on the top, and then there's some space in between, and then there are two thick bands here, and then there's some space here, and then there's a faint band, right? But when you compare the sample here in the next well, it is not exactly the same. It's completely different. And let's go to the next well, which looks similar to this well, which means this is also a hindry digestion. And obviously these three wells, they're not matching with the hindry, but the last well is actually matching with the hindry digestion, right? So I have two samples that were digested with an enzyme called HIN3. And how did I know that? Because I have a positive control here that shows me how the bands look like when you cut the DNA with HIN3. So this is my positive control. And on the other hand, you can see in the ligation, I did not add any controls, right? I'm just showing you this single bands where I'm saying that, all the DNA fragments have been ligated together into a single band, which is good because, I don't see any bands in the bottom, right? So let's say if I started with this sample here, and if I do the ligation reaction with this sample, then if it becomes like this, then obviously it tells me that all these fragments that are in this well have been joined together into a single piece of DNA. Okay, so that's why, even though I don't have a control here, it's okay because I have controls here. I can use all of these as controls because I know how the DNA looks like when it is cut with the enzymes, right? And obviously here, if I join all those fragments, there is only a single band here. So that's how we use these controls. So these agarose gel electrophoresis is something where you use this agarose gel, you load the DNA samples, and you apply the negative charge on this side and positive charge on this side, so that the DNA samples will actually move through the gel from this side to this side. And that happens because DNA is negatively charged. DNA has the sugar phosphate backbone, and the phosphate group, it imparts a lot of negative charge, right? So phosphate, it has a lot of negative charge. So because all the DNA samples are negatively charged, when you apply the negative charge on this side, that will repel the DNA samples so that the DNA samples will actually run towards the positive charge. In doing so, what happens is the DNA has to migrate through the pores that are present in the gel. So when the DNA is moving through the gel, the smaller fragments will move faster because it's easy for them to move through those pores compared to the bigger fragments of DNA. They have a hard time. They cannot move fast because they're big in size. So that's how they get separated, which means the band here that is at the bottom is the smallest piece of DNA within this mixture. And this band on the top is the largest piece of DNA within this mixture. Okay, so this is the agarose gel electrophoresis, and typically we should always add the DNA ladder so that you can compare the size of your bands with the bands in the DNA ladder so that you know the molecular weight of your bands. And you should also add a control, something like this. So this is the molecular biology typical example. Now there's another type of gel electrophoresis called SDS page. So SDS page is used for proteins, and it stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis. So the difference between the agarose gel electrophoresis and the SDS page is that the agarose gel electrophoresis is primarily or mostly used only for the DNA samples. So we don't use the agarose gel electrophoresis for protein samples. But when it comes to the SDS page, this is primarily used for protein samples. Sometimes we use the SDS page for DNA samples, but primarily SDS page is used for protein samples. And the principle is the same. So in this case, it is very hard to see the gel, but I'm pointing out the edge of the gel here. You can see. So this is the gel. This whole thing is the gel. Okay, it's transparent. So compared to the agarose gel, you can see some opaqueness, but when it comes to the SDS phage, it's very thin. It's probably maybe 1 millimeter thick. Okay, so and it's transparent. Because the gel is transparent, it's easy for us to see all these protein bands. So this is sample, one sample, the second sample, third sample. And again, just like how we added the DNA ladder, here we are adding the protein ladder, which means, you see all these bands in the protein ladder. We know the molecular weight of each of these bands in the protein ladder. So that, let's say in your sample, you have a big band here, right? So if you want to know the molecular weight of this band, you can say it's between this much and this much, right? We know the molecular weight of this band and molecular weight of this band. So you can say the molecular weight of your band is between these two molecular weights. Okay, so you can see the protein expressed in the sample. This is a negative control. This is a protein ladder. So what we're trying to show here is, I think for life sciences students, it's easy to understand. We can use the E. coli bacteria for the protein expressions, right? In biotechnology, we often use bacteria to grow and make proteins. So this is somewhat called the recombinant DNA technology. So in recombinant DNA technology, what we do is we take the gene from humans that we are interested in, and we put that gene into bacteria, and we grow the bacteria. So you may ask a question, why use bacteria? Because it is easy and fast to grow the bacteria. So let's say if you want to perform an experiment on human, in order for a human to be born and become 10 years old, it will take you 10 plus years. But if you want to study a gene using bacteria, the bacteria takes probably a few hours. In a matter of 4 to 5 hours, you can get millions and billions of bacterial cells. So that gives us a great advantage. So here in this study, what we're trying to see is the expression of a human protein called glutathione S transferase. It's abbreviated as GST. So here you can see in this protein expression sample where we know that this bacteria is expressing the human protein. So this is a particular band, and the molecular weight is roughly 25. So the size of this band here in the protein ladder is 25 kilodaltons. So, and the size of this human GST is also 25 kilodaltons. And we are using a negative control in which we know that we did not put the gene. Right. So if you did not put the gene into the bacteria, then you cannot expect the protein, right? So this is how you can confirm that, yes, this is the protein that you're looking for because it is absent in the negative control. If I actually did not put this negative control, then, you wouldn't know what to look at, right? There's this big band here, there's this big band here, there's a big band here, there are 3 bands, and then there are multiple small bands, faint bands, right? So it will be very confusing. So that's why if you want to know whether the bacteria have expressed the protein of your interest, then you should compare this with the negative control, and the negative control being the bacteria that did not have the gene of interest or the human gene that you're trying to look for. Okay, so we're using this lane as a negative control, and this is the experiment. And the same thing that we're doing here. We got the expression of this protein here, and this is absent in the negative control, and the protein expression is seen in the sample here. So there's sample 1, 2, and 3. So there are 3 samples in which we see the human protein is expressed, and then we're using 1, 2 negative controls. So now, looking at this gel, I can tell you with full confidence that my experiment worked. And if I didn't have the negative control, first of all, I cannot talk to you with confidence whether my protein is expressed or not, because like I said, there's 1, 2, 3 bands. So I don't know which one it is, right? I hope this is helping you. All right, so let's move on to, so far I have discussed two examples that we commonly use in routine molecular biology. One is the agarose gel electrophoresis, and the other one is the SDS-H, polyacrylamide gel electrophoresis. Now, let me switch gears and talk about the controls that we should be using in microbiology. So, for example, here I'm showing you the patient samples. So, as you can see, this vial here says P1, so that is from patient 1. This says P2, as this is from patient 2. So, basically, what we did was we took this medium, liquid medium, that looks in golden yellow color, and it is transparent, right? So, this is how the medium looks like. And we have taken a patient gastric biopsy, which means, we took a small piece of, the stomach internal layer through the endoscopy. And we added that because we want to see whether we can grow the culture of Helicobacter pylori bacteria that causes gastric ulcers. And as you can see, we're using this as a control because, this is the vial in which we did not add anything. And you can see both patient 1 and patient 2, when we added the biopsy samples, there's plenty of bacteria that is growing. And you can clearly see that the medium is not transparent anymore. It is opaque, right? So if I didn't show you this middle bottle, then how do you know whether this has, bacteria or not, right? What if the media is in this color, right? So that's why you have to, so the qualitative control. So this is a negative control that tells us that, yes, the bacterium is growing in both patient 1 and patient 2 samples. And the same here. As you can see, this is a negative control. This is a better picture. As you can see, there's a light, right? There's plenty of light. As you can clearly see, all these 3 samples, these are all opaque, right? So you cannot see through, but this one is transparent. So this is your negative control. And as you can see here, this bottle is still sealed, which means this is unopened bottle. So in this image, we actually opened the bottle, but here, this bottle is not opened, which means it is a perfect negative control, okay? So there's no more debate whether this is a negative control or not, because, we bought this from this company called High Media. As you can see the label here, High Media, that's the company that sells this medium. So, and we did not open the seal, which means you can 100% trust this as negative control. And looking at the transparency, you can clearly see all these 3 samples have bacteria that is grown from the patient samples. So this is how you have to have controls, because, it's easy to do the experiments, but it is hard to publish the results if you don't have the proper controls. Because when you write a manuscript and submit for publication, the editors and the reviewers, they need something like this. They have to compare your experiment with something like this so that they can determine whether your experiment worked or not, right? So the negative control is very useful here. Now, what we did was we took this bacteria for the patient samples and we prepared the blood agar plates. So in this diagram, I can just show you the tetracycline. So these 2 are antibiotic discs. So there are small filter papers that are dipped in tetracycline solution. Tetracycline is an antibiotic. And here you can see these foams. So each small disc here at the end of this extension has different concentrations. So for example, here this is 0.01 micrograms of amoxicillin. So amoxicillin, ciprofloxacin, and clarithromycin, these are all different types of antibiotics that are commonly prescribed for various bacterial infections. So here, clearly, this white pink color is the Helicobacter pylori that was taken from the patient sample that I showed you here in this slide. So it's growing very fast, right? Compared to the negative control, you can see there's plenty of bacteria. So when we took this bacteria and we spread it onto the plates, you can clearly see there's a lot of bacteria. So this is the negative control. Negative control meaning we did not add any antibiotics here. So we took a simple plate with the blood agar medium, and then we spread the patient sample on the top without any antibiotics. So you can see the bacteria has grown completely all over the plate without any gaps, right? But how do you know that? In order to show that the bacterium is fully growing, you need to show a blood agar plate as it is without any inoculum. Inoculum in the sense adding the bacterium to the plate, right? So this plate, the blood agar plate, without adding anything, looks like this. So compared to this, if you spread bacteria, and the bacteria will grow, and it looks like this. And if you put the antibiotic discs, as you can see here, when you put these two tetracycline discs, there is some dark space here, which indicates that the bacteria did not grow, right? Because of that antibiotic. But when you look at the other place, it's fully grown. So the bacteria is growing everywhere, right? So this is the patient samples. And in the case of amoxicillin, ciprofloxacin, and clarithromycin, you can see the bacteria is growing and there's no dark zones. I mean, yes, there's a dark zone here, but as you can see, there's bacteria inside, which means these patients have bacterium that is resistant to amoxicillin, ciprofloxacin, and clarithromycin, which means this patient cannot be given the antibiotics. You can give tetracycline to this patient because it is showing some potency here because the bacteria is not growing near this tetracycline discs, right? So in order to understand this tetracycline, clarithromycin, ciprofloxacin, and amoxicillin, you need to show a negative control plate and also a plate without the inoculum. So these are your controls. If you don't show these two, then these results do not mean anything. Okay. Now, there are some people, they may not do a lot of lab work, but they may do a lot of computational work. Even in computational work, when you're doing the docking studies, docking means you take a protein and you take a small molecule, something like this, shown in white color in these panels, and you use a software that would, insert the small molecule into the cavities of these proteins and see how they're making these hydrogen bonds. As you can see, these yellow dotted lines, those are hydrogen bonds between the drug molecule and this green colored protein from HIV. Okay, so all the green colored protein is from HIV. So, different molecules make different hydrogen bonds, as a different number of hydrogen bonds. So for example, here in the panel C, we share different panels, A, B, C, D, F, G, H, all the way to L. If you look at panel C here, this part of the drug shows 3 hydrogen bonds, right? There are 3 dotted lines, yellow lines. In panel G, as you can see, this particular drug is making only 2 hydrogen bonds in the same position, right? So, but again, in the panel K, this drug is making 3 hydrogen bonds. So, but how do you know where is your control? So this is our control. This is the molecule. So, that shows a typical profile. Okay. So when you're doing this docking, these are the expected number of hydrogen bonds. So as you can count here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. There's one here, 11. So there are 11 hydrogen bonds. So all these drug molecules that you're docking here, they should show at least 11 hydrogen bonds. Otherwise, they're not good. So that's why we use this control molecule that we already know. So this is a positive control. Okay, so this is for computational biology. So I think in the interest of time, I will just cut cut short here. I think I'll open this up for questions. I guess, you guys can ask me questions if you have any questions. Thank you.
Thank you, Professor. I have a small question.
Yes.
There is something called code breaker.
Okay.
Something called code breaker
Okay.
in CRISPR technology.
Okay.
I could not much follow it.
Okay.
Lot of work is being done on the CRISPR technology. And as I understand from your
Yes.
elaborate lecture about the controls, what exactly
Yeah.
this technology is about, would you mind to explain something to me?
Yeah. So, yeah, go ahead.
CRISPR technology, sir.
Sure. Yes. What happened was previously some scientists have noticed that, when bacterial cells get infected with the viruses, there's a special class of viruses called bacteriophage. So these bacteriophages are viruses that infect only bacteria.
Sir.
So when the scientists have noticed that some bacteria, they have developed immunity. I mean, bacterium is a single cell, right? They don't have immune system. They don't have T cells, B cells, and all those things, right?
Yes.
So they were curious about, how the bacterium gains the immunity against the bacteriophage. So it turns out that the bacterium, when the bacteriophage infects the bacterial cell, the bacteriophage injects its genomic DNA into the bacterial cell. And the bacterial cell will actually take this genomic DNA from the bacteriophage, analyzes it carefully, and then takes a small piece of it as a sample and keeps it in the bacterial cell. So that the next time when the same bacteriophage infects the bacterium, then there is an enzyme, restriction enzyme, that will actually cut the viral DNA so that the virus cannot infect this bacterial cell. So this is basically the observation that was made initially. And then it was the whole CRISPR-Cas9 technology was developed based on this. So what we do is, for example, let's say we have, and so if you have to remove the mutations, or if you have to, repair the mutations, what we can do is we can use the CRISPR-Cas9 package so that the mutant piece of DNA can be replaced by the healthy wild-type DNA. So basically, what you have to understand here is the entire technology works similar to a combination of PCR and restriction digestion. So if you know how PCR works, and if you know how restriction digestion works, and you combine these two concepts together, that is pretty much the CRISPR technology. So in the CRISPR technology, what we do is we design a short RNA molecule, and we call it as guide RNA. It's called gRNA. So the guide RNA acts as a primer, just like the primers that we use in the PCR. So the guide RNA actually goes and binds exactly where you need the mutation repair. So for example, in chromosome number 4, you have a gene that has a mutation. So you design your guide RNA so that the guide RNA will actually go and bind to the DNA exactly in the chromosome number 4. And this guide RNA has a special secondary structure, this stem loop structure, that is specifically recognized by a restriction enzyme called Cas9. So Cas9 is basically a restriction enzyme like, EcoR1, INTRI, MAMH1, and all these. So then, the Cas9 will actually go and recognize this secondary structure of the guide RNA. It will bind to that, and then it will start cutting the DNA.
Sure.
So that's basically the CRISPR-Cas9 technology. But then you may ask me a question, you're cutting the DNA and how can you leave it, right? That's a problem. You cannot just cut the DNA both the strands and leave it open, right? That would be a big problem.
Yes.
So the biggest problem or the biggest challenge for the CRISPR to work is the cells have DNA double-strand damage repair system. This DNA double-strand damage system can happen in various different ways. You know, the simplest way would be homologous recombination that involves the formation of holiday junctions and etc.
Sir.
So when the CRISPR-Cas9 cuts the double-stranded DNA exactly where you have the mutations, then that mutant piece is removed, or it comes out of that chromosome number 4. Then immediately the cellular DNA repair mechanism will start the homologous recombination based DNA repair, so that you have a wild type gene that is built into that chromosome number 4. So in the CRISPR-Cas9 technology, the first step is something that we will do, and the second step is done by the cell by itself. The repair part is done by the cell by itself. I hope that makes sense.
Yes, sir. Is it a kind of recombinant DNA technology?
No, this is homologous recombination. I would say the best example would be the synopsis or the crossing over that happens in meiosis. The cell division, typically we have two types of cell divisions, right? Mitosis and meiosis.
Yes.
So in meiosis, there's something called crossing over. The pieces of chromosome from one chromosome to another chromosome, they get crossed over, right? So that is the best example for the homologous recombination. Recombinant DNA technology is a technology where you're taking a gene from humans and you're putting it into the bacteria so that the bacteria can make the protein from that gene. So that's recombinant DNA technology.
This kind of gene editing, any unintended consequences for human beings, sir, on ethical side?
Well, I mean, I think most of the countries are not really confident about this yet. But there are scientific evidences that prove that CRISPR technology is pretty solid. But then there are also some papers in the literature that say, it may go wrong. So there are a few, not too many, but just a few examples where if the Cas9 enzyme does not cut properly, or what if it cuts at multiple sites instead of just one site? I mean, you have to understand it all depends on the specificity of the guide RNA that you're designing. If your guide RNA is highly specific to this sequence that you're targeting, then you should be fine. But if we make any mistakes, then that guide RNA can bind anywhere in the genome, right? So wherever the guide RNA binds, then the Cas9 will go and cut it, right? So that's
Yes.
There are drawbacks, that's for sure.
Any effect on eugenics? Extra type of things, people may create monsters also by using this technology.
Well, I mean, there are ethics solidly placed. I guess one of the reasons could be that. I don't think we can actually generate monsters using CRISPR-Cas9, but I cannot deny that claim. There might be a possibility. Yes, there are ethics. There are very strong ethics. Usually, when you do this kind of experiments, we need the institutional ethics review committee's approval, and we have to explain everything. What if something goes wrong? What is the plan B and what is plan C? So we have to explain all those in our proposals when we apply for these grants and funding, etc.
We do need safety controls for these kinds of experiments.
Oh yeah, absolutely.
Thank you, sir. Thank you.
You're welcome.
This mechanism which you have explained is like the drug resistance that is developed by the bacteria. Is it like that, sir?
You can use this for anything. So you can basically repair the mutated genes. You can use it in bacteria, you can use it in viruses, you can use it in animals, plants, or humans, anyway.
Yeah, in the Second World War time, when the penicillin was a real miracle drug, subsequently now the usage of penicillin has been reduced. Is it because of its overuse and then the bacterium has developed some kind of reaction? Am I making any sense out of what I'm asking, sir?
I'm unfortunately, I have to answer your question in two different ways. So, being a scientist myself, I should say that yes, there are definitely a lot of multi-drug resistant bacteria, and obviously they have gained resistance because of repeated usage of this penicillin. But on the other hand, just as a civilian, as a citizen of India, I can say, these days, or actually from a long time, people don't usually go to the hospitals. They don't see the doctors. They can just walk up to a medical store and they say, I'm not feeling well, I have a fever. And then the medical store actually gives them an antibiotic. And obviously, the more antibiotics you use, the more drug resistance will be caused because you may not be using the proper antibiotics for the proper reason. So, for example, you may have a gram-negative infection and you may be using an antibiotic that is targeting the gram-positive bacteria. The medical store guy doesn't know this. So that's why it's a good idea to go to a doctor and have the infections. But this is from a general civilian point of view, not as a scientist.
Thank you, sir.
Thank you, sir. Dr. Ravikiran, sir. You gave an extraordinary speech, extraordinary session that we have enjoyed it a lot. Though we are not belongs to these departments, that we can understand how this mechanized world is involved into this kind of emergencies in science and technologies. Thank you very much, sir. That how to control all these things in being as a student and scientist in all these different areas that you have given an elaborate explanation about all this. Thank you very much, sir. And it's time for me to propose the vote of thanks. From the organizing desk, from the Department of Biotechnology, Physics, Electronics, History, Chemistry, and association with IQSC, that we are proposing vote of thanks to all these important persons, those who had given and contributed so much for this program. I start with APV Apparao, sir, principal of our college, Dr. C. Satyanarayan Rao, sir, secretary and correspondent of our college, Mr. R.V. Subbarao, sir, retired scientist, academic and research advisor, Sri Vayan College, and special thanks to Dr. Ravikiran, sir. Thank you very much for your explanation and presentation. And that's all from the presentation party. And now I request all the participants who are being online, we are sending Google Forms and feedback forms in a link. So please do and please give the genuine feedback. Thank you. This is Mr. Abhilash signing off.